So this is the answer booklet for DNA and Inheritance 4.3 Additional Biology. Again, here is the knowledge organiser, which you should have used to help you answer the questions. And the first question, name two scientists whose work led to the discovery of the structure of DNA. It was Watson and Crick. A section of a single strand of DNA has the following sequence of bases. What are the complementary sequence of the bases that are shown above? This is just remembering that A binds to T and C binds to G. So TAG, ACA, TGT, So the maximum number of amino acids that can be coded for the sequence. Number of amino acids are coded by the triplet code, which 3 bases. So these 3 bases are coded for one amino acid, for a second amino acid, for a third amino acid. That's on its own. We need 2 more to make our triplet code. So it's a maximum of 3. A DNA molecule consists of 2 coiled strands. When cells divide, the 2 strands in the DNA separate. The bases of each strand pair with complementary bases to make new DNA. Complete the diagram below to show the sequence of the complementary bases that would appear in this DNA molecule by drawing the shapes of the 4 missing bases and label each of them with their correct names. So we're looking at that it fits in, a bit like a jigsaw. So you can see I've got the 4 drawings. And then we have to label them. So we know, because they have told us, that cytosine is this shape, and I've just drawn it there, so we have cytosine. They've also told us adenine, so I've got adenine, and then it's remembering that adenine binds with thymine, so that has to be thymine, and then cytosine binds with guanine, so that has to be guanine. Describe how a section of DNA can control the formation of a protein. It's the triplet code. So 3 bases, code for a specific amino acid, and they then join together to form proteins. Outline the basic structure of DNA and explain the meaning of importance of the genetic code. I don't know why this question is not answered. I do apologise. I will go through it quickly now. I'll zoom in so you can write on it. So the first thing I want to say is that it's a double helix. As it's a double helix, we mean that it's 2 strands twisted together to form a helix. The strands are made up of alternating sugar phosphates. Alternating sugar and phosphate. Then we can say that there are 4 bases. You need to name the bases and you need to be able to spell them correctly. So we have adenine, thymine, cytosine and guanine. With that then, we could do the complementary pairing. Complementary pairing, and we know that adenine binds with thymine and cytosine binds with guanine. Then looking at the production of proteins, we have to mention the triplet code. And the triplet code is 3 bases, code for an amino acid, which makes proteins. Proteins. Okay, next question. The photograph below shows a scientist who worked on the structure of DNA in the 1950s. How was the structure of DNA discovered? Choose one of the following statements to answer the question, and the answer is B, by many scientists using a number of different techniques. The diagram below shows part of a DNA molecule. Complete the sentences about DNA using some of the words below. DNA is made up of 2 long chains of alternating phosphate and sugar. It doesn't matter which way around you do it. Which are joined by the bases A, T, C and G. DNA is twisted to form a double helix. Why is the order of the molecules A, T, C and G in DNA important in the production of proteins? And it's because 3 bases code for a specific amino acid. The diagram below shows part of a DNA molecule with a small section in detail. Molecules known as A, T, C and G join the chains of sugar phosphate shown in the diagram. What is the general name for these molecules? The answer are they are bases. Fill in the two missing letters on the dotted line, so they are here. So we know that adenine binds with thymine and cytosine binds with guanine. Which part of a living cell contains DNA is the nucleus. And then the diagram shows DNA as a ladder-like structure. How is the shape different in a living cell? It's a double helix. The diagram below shows a small section of DNA. We've got 5 bases are shown in one strand of DNA. Fill in 5 missing letters from the bases on the other strand. So we've got A to T, C and G, A and T, T and A. Just remembering the complementary base pairing. Complete the label on the diagram by writing the missing word on the dotted line. Long chain of sugar and phosphate molecules. Insulin is a protein. The structure of insulin is shown below. Underline the name of the small molecules labelled Z, which make up the protein. So we've got amino acids, they make proteins. Why are the bases in DNA important in building proteins from these small molecules? That we have 3 bases code for specific amino acids, which then make proteins. Explain what's meant by the term genetic profiling. It is separating DNA into bands, and then 2 uses for criminal cases, solving crimes, or paternity tests. In 1850s, it was thought that there were 2 different species of elephants living on Earth. In the 1950s, some scientists suggested that these 3 species of elephants existed. These were the Indian elephant, the African plains elephant, and the African forest elephant. In 2011, an analysis of the chemical structure of chromosomes of African, Indian, and fossil elephants were carried out. This provided evidence that the African and Indian elephants had a common ancestor 2.5 million years ago and confirmed that 3 species of elephants exist today. Name the chemical in the chromosomes that was analysed. The answer is DNA. The units made of this chemical, which make up chromosomes. So they are our genes. Sorry, I don't know why that question is missing. And the diagram below is a simplified family tree showing the evolution of elephants and their ancestors. Each letter represents a species. Refer to all the information given opposite, answer the following questions. Which letter or letters in the diagram opposite represent a common ancestor to more than one species? That would be E. An extinct species would also be E because it's 5 million years ago. The Aphaeus indictus would be A, and the 2 closely related species would be B and C. PKU is an inherited disease caused by a recessive allele. PKU results in damage to the nervous system in the early years of life. Key use letters N's to represent PKU. A couple neither whom suffer from PKU have a child who is found to have PKU. State the genotype. So the genotype is the set of alleles the organism has. So it can either be with the little n, big N, big N, big N, little n, or little n, little n. Can't be little n, because they haven't got PKU. And to have PKU, you need 2 little n's. So they have to be either big N, or big N, little n. If they've both got a child who has PKU, they have to be carriers of it, both of them. So it makes them big N, little n. Complete the Punnett square. So how I've got big N, little n, and there's my outcome. Place a circle in the Punnett square on the genotype of the child suffering. So it's little n, because it's caused by a recessive allele. From your Punnett square, what's the probability of a child not having PKU? So 3 out of 4, 75%. In mice, the allele for black fur B is dominant over the allele for white fur little b. 2 heterozygous, so there's a key word there for us. Black mice are mated together. So heterozygous, the alleles are different. They produced a litter of 8 young mice. Which of the following results would you expect in the litter? So we're looking at a litter of 8. So we'd have 6 black and 2 white. And our Punnett square shows it. So we've got heterozygous, the big B, little b for different. And if I look at it, I have 1 that is white out of a litter of 4. So I'm going to double it then for our litter of 8. So I'll have 2 white, 6 black. The sex of the mice is determined by which sex chromosomes are present. State the sex chromosomes presented by the body cells. So for a male, it's XY. For a female, it's XX. Complete the Punnett square below to show the expected sex chromosomes of the offspring of the litter. So as you can see, you've got XX, XY, and it works out to be 50%. Literally half and half. A grey-bodied fruit fly was mated with a black-bodied fruit fly. All F1 offspring were grey-bodied. Use the letters A to represent the alleles of 2 different body colours. Complete the Punnett square below to show the offspring produced when the mating occurred between the grey-bodied and black-bodied. Right, the question gives us a lot of information. So, grey-bodied was mated with black-bodied, and all the offspring were grey-bodied. So we know the grey-bodied is dominant and the black body is recessive. So the black, they must have 2. So we know he's little a, little a. This grey-bodied though could either be big A or big A, little a, to work out which one he needs to be. If all of the offspring were grey-bodied, he has to be homozygous to pass on the dominant allele to every single one of his offspring, which is then shown here. Big A, little a for our black-bodied. And we have all heterozygous offspring, but they're all grey-bodied. Complete the Punnett square below to show the offspring produced when 2 of the F1 offspring are selfed. So taken from our last question, we have all heterozygous offspring. So this is the exact same. So little a, big A, little a, big A, little a, and we have our results. What's the ratio of the phenotypes? So another key word. Phenotypes is the physical characteristics. So essentially like eye colour being blue. And then name, so we have 3 to 1 because I've got 1 dominant allele, 1 dominant allele, 1 dominant allele here. So they would have grey-bodied, and then I have 1 that is recessive, homozygous, so they would be black-bodied. And then the scientist who worked on it was Gregor Mendel, so we can just put Mendel down. Until the 1960s, turkeys bred for meat on a turkey farm all had black feathers. Then mutant pure white feathered turkeys were hatched in a turkey farm. Turkey farmers used the mutant turkey to produce all white feathered flocks of turkeys. Almost all turkeys sold in supermarkets today are of the white feathered variety. What type of genetic variation is shown by the feather colour in the turkeys? Don't know I've got genetic there because the answer would be discontinuous. Why is it not possible to breed black feathered turkeys from white feathered turkeys? Because no black allele in white turkeys are homozygous recessive. So they only have, for white turkey, they need to be little b, little b. The second they get a black allele, they become a black turkey. Research into breeding turkeys for various characteristics is taking place. Scientists have attempted to genetically modify some varieties to increase meat production. The genetic profiles of the 5 turkeys, A, B, C, D, and E, are shown below. The turkeys are shown by the letters A to E. The positions of the bands of the profiles are shown by numbers 1 to 19. 2 of the turkeys were hatched from the same egg, using their profiles to identify them. So the answer is A and C. And the reason being that they have the exact same genetic makeup. Okay, their banding are the exact same. The absence of a band at position 10 leads to an inherited disease caused by the altered DNA. Identify the 2 turkeys with this inherited disease and suggest why it would not be a good idea to clone these 2 turkeys. So at number 10, if I change my colour highlighter, at 10, the 2 that are missing number 10 is D and E. And then not to make them together because all the clones would have the inherited disease. All the turkeys except D are white feathered. Which number shows the position of the mutated colour? So let me change it. And if we look, it's position number 3 because D is missing that band, whereas all the rest have it.